Hi, my name is Sri Kosuri. I'm the co-founder and CEO of Octant, and I'm also an associate professor at UCLA. I wanted to thank the organizers for giving me a chance for me to talk today. I think now more than ever, especially with the results from Cas14 and AlphaFold, really show that AI is here to stay in biology. But I also think this is just the beginning. What I'm going to talk to you about today is this confluence of technologies like DNA synthesis, genome editing, functional genomics, high throughput chemistry, when brought together through computation, can do high throughput biology at scales we only dreamed about a few years ago. And importantly, I think we're in a new age, especially in the coming decade, where experiments are no longer going to be the limiting thing, that we can build and conduct tens of thousands of experiments routinely. And that's going to have a bunch of challenges that are going to stretch individuals like ourselves, where we're trying to design biology and design experiments at the scale to do discovery biology. But more broadly, this kind of virtuous cycle of design of experiments or essentially generative biology using I think is going to be one of the most exciting things that in the coming decade. So I wanted to start off by talking about how I have always thought about synthetic biology as a foundation for for discovery in general in biology. And it comes down to this idea of models and simulations, and we normally uh, think of those models and simulations of biological processes through the lens of computation. But for a long time, that's not always been the case. So what I'm showing you here is the Mississippi River Basin model, first built by the Army Corps of Engineers soon after the World War II by actually German prisoners of war. It's a 1 to 2000 model horizontally, 1 to 100 vertically of the Mississippi River Delta. And the reason why they built this is that there were a series of floods during this time that caused a lot of damage to farmlands, to crops. And what they wanted to do was simulate those flooding events in order to understand how they could put interventions or dams into different parts of the river in order to avoid severe flooding of these lands. And it was ridiculously accurate. Of course, we don't want to do this broadly just because this took a ton of effort and time and free labor. But it's something that's really interesting. It's a physical model of a system of which it's meant to simulate. It's just a smaller version of that. And then you can run multiple simulations on of floods or rains and different points of entry of that water in ways that let you make predictive interventions in order to engineer solutions. And so this is a great simulator. I think when we think about this in biology, this is also nothing new. In biology, we engineer cells all the time, and we've been doing that since the dawn of molecular biology to act as models to understand if I change something, what are the consequences of that? And the very beginnings of this were the very beginnings in the dawn of molecular biology. So this experiment here is Marshall Nirenberg in Matai's lab in a little known lab at the time at the NIH, where there was this big problem in trying to discover the genetic code. And the basics of that are the genetic code, 64 codons, 20 amino acids, which we know now, but people didn't know what that code was at the time. And what Nirenberg said was like, if we can use polynucleotide synthesis, or the synthesis of mRNA, to make polyuridine initially, then we can see what it encodes by putting it into a cell extract and seeing what protein comes out. And what they showed here is that encoded phenylalanine. And then their lab, Phil Leader's lab, Govind Karan's lab, through the systematic production of triplet nucleotides, showed that you can basically break the genetic code, and they did this systematically for all 64 codons, and use synthesis as a way to do discovery. These weren't natural sequences. It wasn't an inference that discovered the genetic code. It was just pure brute force synthesis. So in this way, the cell extract was it was a physical model of the biological system they were trying to understand. And one of the things that you can use synthesis for is as a way to simulate across that model. a lot of what we think about at Octane is really bringing that to that type of thinking, of using the cell as your physical model and using the ability to do DNA synthesis and editing as a way to modulate or simulate on that model, as a way to do discovery biology en masse. And what that set of technologies is a suite of technologies, including DNA synthesis, genetic editing, DNA sequencing is a readout technology, and I'll talk to you about that in a second. And bringing those all together through genomics and computation in order to do experiments at scales that kind of reach our genomic problems today. So uh, you know, I'll talk to you a little bit about the base sets of technologies that make these types of things possible. The first one, which I'll largely skip over because there's a suite of companies and technologies and papers out there talking about DNA synthesis technologies. And a lot of that drop in price and increase in scale has come through basically DNA printers, places like Agilent Technologies and Twist Biosciences that make millions of oligonucleotides on glass surfaces or silicon surfaces to basically design spec. And so you can build millions of these sequences that you can program on a computer and harvest them and introduce them into cells. The second part of the thing that might be a little less known are things called multiplex assays. And multiplex assays, all that really means is that you do an experiment. So you introduce a lot of these genetic constructs that you've designed into cells, where each cell maybe gets one of these things. And then you do some assay that separates the function of all of those genes, depending on the function you're looking for. And usually you read that out through a process of next-gen sequencing. And we do that in many ways. My lab over the years has done a bunch of these kinds of multiplex assays looking at all sorts of functions. At Octane, what we do is to really scale these types of approaches in human cells to do drug discovery. The way we can think about that is that usually we're trying to uncover or understand the function of a particular gene in a cell, in a human cell line. And so we engineer that receptor into that cell and the signal transduction pathways and or functional pathways that let us understand the function of that gene in response to different chemicals or endogenous agonists or just the natural gene's function. These are usually called reporters. And these kinds of reporters can be built and in fact are a big part of drug discovery today and for a long time. So the classic what's called luciferase assays or GFP assays basically shine a piece of light or wavelength of light if the receptor is activated. And say I make also a second experiment with a mutant of that receptor, I'd have to do a second experiment. What we do is add a barcode to the end of those reporters. And what all that means is that like it's a series of sequences, ATC or G, or say barcode 1 could be ATCG, barcode 2 could be GCTA. We use next generation sequencing as a way to read out those differences. So the number of copies of RNA that we see of that barcode is the activation level, and the sequence of that barcode tells us which particular experiment we're trying to do in that mixed dish of cells. So then we combine all those cells together in a way that lets us do what are called multiplexed experiments. And one of those experiments was around a class of receptors called G protein coupled receptors, and in particular, a subset of those called olfactory receptors. So GPCR, there's about 800 of them in the human genome, half of which are responsible for our sense of smell and taste. The smell ones are called olfactory receptors. The other half are responsible for the targets ultimately of about a third of FDA approved drugs and about 60% of prescriptions and basically most natural products and illicit drugs. So we were interested in trying to build a multiplex assay of how chemicals interact with these receptors that sit on the surface of cells. And in large part, the olfactory receptors, the ones that are responsible for the sense of smell, are largely orphan. What that means is we don't even know what they bind or react to. And so the way we did this is we engineered these receptors. We have to engineer a bunch of pathways in the cell that lets us like express those things functionally. And then if they get activated, turn on one of these barcodes. In my academic lab at the time, Eric Jones, and Rishi Jaju led this up. They kind of took a set of 40 G protein coupled receptors. In fact, these were murine or mouse olfactory receptors. And they introduced them into cells. They mixed all those cells into one well of a 96 well plate. And then they just started doing small molecule screens. So all we're doing is measuring the barcode expression on each of these wells and doing small molecule screens across them. What we started seeing is that we could start de-orphanizing many of these receptors by doing small molecule screens of odorants. And all that means is that we found an active ligand towards those receptors that were specific towards particular molecules. And so you can see some of that data on the right, and you start picking out patterns of different receptors and the types of moieties they're starting to look at. And of course, as you probably know, and you're probably here looking at these kinds of things, you can apply kind of standard machine learning tools. This was, the variational autoencoders that from Alan Asparuh-Gutsik's lab, where you can start picking out similar types of molecules that are responding to different types of receptors to understand their specificities. We at Octant, while this is interesting for flavors and fragrances and things like that, this is also pretty interesting for the processes of drug development. And so we've started scaling this a couple of years ago. And at this point, it pretty much operationalized and scaled this process to the point of where pretty much weekly we run, this is about 25 to 30% of a normal run, where you get data sets where in a single well, you're doing hundreds of barcodes, or about 100 barcodes per well in 384 well plates. And we do this in a pretty automated or simple process. It's not necessarily completely automated, where we do anywhere between 8 and 16 plates on a normal week's run. And we're trying to triple that scale over the course of the next several months. So this is kind of a week in, week out process. We do this quite routinely. The analyses are automated. And so what that lets us do is do large chemical libraries and screen them against large receptor libraries all at once in a semi-automated platform that lets us kind of pull from this on a routine basis. And we use this not only to pan these kinds of things, but also in the process of synthetic biology development of reporters for different types of genes. And so it turns into not only our kind of analysis pipeline, but also our pipeline to basically engineer new cells to look at different types of genes and different types of function. We've been scaling this internally at Octant. We have like a core, essentially, or a platform development group that combines high throughput synthetic biology, cellular engineering, high throughput screening, and the analytics to basically turn this process routinely. And we're continuing to expand that internally. One of the internal projects we have is to really look at how we engineer kind of small molecules that are known as polypharmacological, or drugs, and that have a lot of where they hit many targets at once, and they're also known as dirty drugs, or that they hit particular receptors in particular ways, and meaning they turn on one pathway over another. This is often called bias or functional selectivity. The classic set of receptors where this occurs are these kind of central small molecule signaling receptors that are found all over the body, things like acetylcholine, adrenaline, dopamine, serotonin. And we've actually added histamine since then, set of about 35 receptors that are the target of a countless number of drugs and natural products. And there's tons of known polypharmacy and unknown polypharmacy and bias. So there's a lot of challenging biology here. There's a lot of different signaling pathways involved here. And what we've been doing is systematically decomposing those in order to find conditions that let us screen these things in individual wells across all of these receptors at once. And what's interesting is that the best-in-class drugs for a lot of these receptors, so things like atypical antipsychotics like clozapine that you see here, really interact with a bunch of these receptors at the same time. So they are agonists, partial agonists, inverse agonists to these different receptor classes at once. And a lot of what we try to uncover are two things. One, how do we identify the sets of polypharmacological signals that we think are important to target for a particular phenotypic outcome? And then secondly, how do we build molecules to be more rationally targeted toward those things? So the second part of that is a high throughput synthesis platform we've also been building over the past year. And the basic idea behind the synthesis platform is that we kind of know the starting points of molecules to hit for these types of receptors. But the thing we don't know how to program are things like polypharmacy and bias. And so what we do is take existing chemical cores and basically decorating them with small fragments. So it's basically a high throughput fragment-based chemistry approach where we can do simple one-step reactions in acoustic microfluidic devices and to build 5,000 to 10,000 chemicals at a time that we can drop onto our platform kind of multiplex analysis technology. And we do this in a way that lets us basically kind of iterate through this cycle quickly. So this has been really exciting. We've kind of just been talking about this over the last month or so. That we kind of just gotten this working over this last summer, and we just started it this year. And obviously, because of COVID, things have been slowed down. But I've been super impressed with this now cycle of being able to build lead compounds towards the things that we've been particularly excited about. The other type of experiments that we do are these kind of scaling up of the process of barcoding. So this is not in an individual well at this point, but what we can do is make hundreds of thousands of barcoded cell lines all in a single dish, each one looking at a particular mutation of these types of receptors. And what we can do is screen different drugs against these mutational libraries to understand how mutation affects the interaction of drugs or perhaps the function of the particular genes. And we use that also for a bunch of different things internally. That includes trying to interpret human genetics. So like how does mutation to druggable receptors affect things like chemical pharmacogenomics or even disease manifestation. We also use this for looking at processes like structure-activity relationships and signaling bias, and finally, as ways to think about targeted molecules towards particular mutations. We've started bringing all of these together, these kinds of processes to, as I mentioned, think about multi-target drug discovery or polypharmacy and how to engineer it rationally. Things like molecular engineering, small molecule molecular engineering, as I talked about, and then finally using these kind of deep scans as a way to kind of decode these large-scale biobank data sets that relate genotype to function. The thing that we can provide is help collapse the variation seen in a particular gene down to biochemical function that can later be regressed on phenotype. And so these are all very exciting things for us, and we're really excited that we might be able to take these kinds of biological technologies that are bringing biology into just like a really genomic age of being able to do experiments at the scale of genomics. And ultimately, what we're interested in often is in each well of a 384 well plate, there's like, on order 20,000 cells, and there's on order 20,000 genes, and it's most manifest, can we start understanding the interaction of molecules, whether that be flavors and fragrances or food or nutritional products, all the way to drugs, and how they interact rationally with the 20,000 potential targets that they might hit in the human genome. So we think this will be really interesting and valuable for a long time, but the thing we're most interested in over the next couple of years, and I think most relevant to this group, is that this is not just true of our problem, and it's not just true of biological systems in general, but like or small molecules in general. It's that for many problems in biology and drug development, we're getting to this age over the last couple of years, that's all it's been, that we can build and test tens of thousands of designed chemicals or regulatory elements or synthetic proteins from near infinite complexity. If you think about this, like a 100 amino acid protein has 20 to the 100th possibilities. You will never simulate that many. You can never actually build all of those. It's more atoms in the known universe, right? So the real question is, how do we pick 10,000 of those at a time to best get us at function, to best get us at intervention, to best get us at understanding, and to best get us at design? I think that is fundamentally an AI problem, or it's fundamentally a science problem of design of experiments. But I think there's a fascinating number of ways to start attacking that. And I think there is no other place other than biology where you can specify 10,000 physical systems on a weekly basis for pretty cheap and see the functional consequences of all those things in the real world, in this world of atoms and not bits. And that's super exciting. I think it's a way for us as a society to really take these kind of new algorithms that are coming out in design and generative models for a spin in ways that have real consequences to society. So that's super exciting. I'm excited to see what happens with this field in the coming years. I just wanted to thank a few people from my academic group that is largely shut down now at UCLA, but really seeded a bunch of the work that's turned into Octant. Octant now is a set of this 13 at least PhD scientists and a bunch of programmers that are just starting and more recent grads that are RAs, but we're a pretty small group and able to do these kind of very large scale experiments, and that's exciting. And I think we're only going to see that continue over time. So with that, thanks so much. Thanks for the invitation, and excited to see the rest of the talks here today. Thanks.